Dear students, very good afternoon. Hope you all are fine. Today's our offline class is in continuation with yesterday's offline class that was about mutation. We discussed in the previous class that mutations are of three types: gene mutation, chromosomal aberrations, genomatic mutation. Out of these three types, we have discussed gene mutation and half of chromosomal aberrations. Chromosomal aberrations, the important aberrations are deletion, duplication, translocation, inversion. Out of these important aberrations, we have discussed deletion and duplication in our yesterday's offline class. Today we shall be discussing about translocation and inversion as well as genomatic mutation. Translocation, it involves shifting of a part of one chromosome to another non-homologous chromosome, so new recombinant chromosomes are formed, as this induces faulty pairing of chromosomes during meiosis. An important class of translocation having evolutionary significance is known as reciprocal translocation or segmental interchanges, which involves mutual exchange of chromosome segments between non-homologous chromosomes, that is, illegitimate crossing over. Chronic myelogenous leukemia (CML) occurs due to translocation of segment of long arm from chromosome 22 to chromosome 9. Chromosome 22 is called Philadelphia chromosome. Now, inversion. Inversion in linear order of genes by rotation of a section of chromosome by 180 degree. Inversion occurs frequently in Drosophila as a result of X-ray irradiation. They may be of two types. Number one, paracentric, and number two, pericentric. Paracentric, inversion without involving centromere. Inverted segment does not carry centromere. Now pericentric, it is inversion involving centromere. Now third type of mutation, that is genomatic mutation. It is change in chromosome number that bring about visible effects on the phenotype. It is of two types. Number one, aneuploidy, and number two, euploidy. Now let's take first aneuploidy. In aneuploidy, any change in number of chromosomes in an organism would be different than the multiple of basic set of chromosomes. It commonly arises due to non-disjunction, that is absence of separation of two homologous chromosomes during cell division of the two chromosomes of homologous pair, so that one gamete comes to have an extra chromosome n plus 1, while the Other becomes deficient in one chromosome, that is n minus 1. This is the diagrammatic representation. We see the gametes n minus 1 and n plus 1. Fusion of these gametes n minus 1 or n plus 1 with normal gametes n give rise to different types of aneuploids. A table gives various types of aneuploids, gametic fusion and type of aneuploid. Number first, n into n minus 1, type of aneuploid, that is 2n minus 1, that is monosomic condition. Number second, n minus 1 into n minus 1, that is 2n minus 2, type of aneuploid is nullisomic condition. Number third, n into n plus 1, type of aneuploid is 2n plus 1, that is trisomic condition. And number 4, n plus 1 into n plus 1, type of aneuploid is 2n plus 2, that is tetrasomic condition. First one, it is normal 2n. Second diagram, it is monosomic 2n minus 1. You can see 1 is absent. Nullisomic, it is 2n minus 2. Trisomic 2n plus 1. And tetrasomic, it is 2n plus 2. Examples of aneuploidy. Number 1, trisomy. Downer syndrome, Killenfilter syndrome. And number second is monosomy. It includes Turner syndrome. Now, type B of genomatic mutation, that is euploidy. In euploidy, any change in the number of chromosomes is the multiple of the number of chromosomes in a basic set, or it occurs due to variation in one or more haploid sets of chromosomes. Accordingly, these may be haploidy or polyploidy. Now, haploidy means one set of chromosomes. Haploids are better for mutation experimental studies because all mutations, either dominant or recessive, can express immediately in them, as there is only one allele of each gene present in each cell. Polyploidy means more than two sets of chromosomes. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is called as polyploidy. It is often seen in plants. In case of animals, polyploidy usually results in sterility. Therefore, polyploidy is rare in animals. Polyploidy fall into two major categories, that is, one is autopolyploidy and second is allopolyploidy. This was all about today's offline class. Thanks.